All right, well, welcome everybody to Fern Spaulding's thesis defense. I just want to say a couple words to introduce Fern. Fern began with us in my lab as an undergraduate here at UAF, and she got her she got her master's degree in biology here again at UAF, and Fern's been a really integral part of our lab in all respects, specimen collection in the field, preparation. Fern's a phenomenal specimen preparator, and we've enjoyed having her. And as she's done, she's carried on a tradition that we've had here for a while. People get really excited about birds when they work in our lab, undergraduates, and frequently we're able to tempt the best ones to stay on and tear into some depth into some of the research questions that the collection presents the opportunities to follow through on. And so Fern has spent almost exactly now 2.5 years with us as a master's student, and her defense today is about looking at the genomics of trans-Beringian birds in two really interesting contexts. And with that, I will turn it over to Fern and let her tell you about her work.
Excellent. Well, thank you, Kevin. So everyone, thank you for coming to my thesis defense today. I really appreciate it. And my thesis defense is using ultra-conserved elements to estimate gene flow between Asian and North American avian taxa here at the University of Alaska Museum at the University of Alaska Fairbanks. So first, what I'm going to do is I'm going to go through a little bit of a background into the work that I actually do at the museum. Then we're going to discuss my chapter 1, and the title of my chapter 1 is Population Genomics and for Different Modes of Divergence and Speciation with Gene Flow in the Green-winged Teal Duck Complex. Then we're going to go through and discuss my chapter 2, which the title is Estimating Movement Rates Between Eurasian and North American Birds that are Vectors of Avian Influenza. And then at the very end, we'll have time for questions and answers. So with that, let's go into a little bit of background into the work that I actually do at the museum. So the work that I do at the museum and the work that I do for my master's degree is specimen-based research at the University of Alaska Museum. Specimen-based research itself allows us to ask lots of different questions, but specifically regarding my study, I can ask questions about modes of divergence and speciation, phylogenetic relationships, how much gene flow is occurring, and even the practical applications of population genomic methods. And here on the right-hand side, we acquire specimens either through field collection or even salvage through donations to the museum. And then these specimens that we acquire are prepped into a vouchered study skin that are used to answer these questions that we're interested in. So a brief overview of my work, and specifically an overview of my chapter 1. My first chapter examines the green-winged teal and yellow-billed teal duck complex. This group previously has demonstrated unresolved phylogenetic relationships in both mitochondrial and nuclear DNA. And so bringing a genomic scale data set to this complex, I ask 3 questions. First, what are the phylogenetic relationships of the group? How much gene flow is occurring? And what are the modes of divergence and speciation occurring in this group? Continuing on with a brief overview of my second chapter, I aim to apply population genomic methods to estimate levels of gene flow in vector species of avian influenza that are also common subsistence species in the diet of many Alaskans. And I aim to improve estimates of intercontinental avian influenza host movements in these subsistence species to better understand the natural host virus movement transmission landscape in Beringia. And understanding the relative risk rankings of these avian influenza vector species should help in modeling, monitoring, and mitigating the impacts of intercontinental host and avian influenza movements. So now we're going to jump into my chapter 1. And again, my chapter 1 title is Population Genomics and for Different Modes of Divergence and Speciation with Gene Flow in the Green-winged Teal Duck Complex. We're going to go through a bit of background from my chapter 1 first, talking about 3 main points. First, what are the processes of speciation and divergence? Second, what modes of speciation are common in birds? And third, why is Beringia an excellent place to study speciation? So first, what are the processes of speciation and divergence? Speciation itself is defined as the process of evolutionary divergence that results in distinct groups of organisms that are recognized as species. This process has historically been categorized into three main modes, each of which is based on spatial relationships between the diverging populations. These three main modes are sympatric speciation, parapatric speciation, and allopatric speciation. So let's define these three main modes of speciation. First, sympatric speciation is the evolution of reproductive isolation while ranges of populations overlap. Parapatric speciation is the origin of new species in which gene flow occurs across a spatially restricted contact zone, such that only a fraction of each population has a high probability of interacting with other immigrants. And finally, allopatric speciation is divergence of populations that are geographically separated from each other with no movement of individuals between them. Effectively, gene flow is equal to 0 in this mode of speciation. So this is a visual representation of these three modes of speciation, where speciation itself exists on a continuum, where the endpoints of that continuum are either allopatric speciation, as shown down here, where the two populations are geographically isolated and there is no overlap between these two populations. And at the other end of the continuum, sympatric speciation occurs where the two ranges of the populations do overlap. And in the middle, we have parapatric speciation where there's this narrow zone of contact between these two populations. So continuing on with processes of speciation and divergence, what modes of speciation are common in general? This is actually a very difficult question to answer, but some general things is that the drivers of divergence for allopatric speciation include geographic isolation, low dispersal rates, and complete isolation. Likewise, parapatric speciation and sympatric speciation, drivers of divergence can be ecological, environmental, and behavioral factors. So what modes of speciation are common in birds? Historically, it has been thought that allopatric speciation was the predominant mode of speciation. However, with the availability of genomic data, the concept of speciation with gene flow has become much more common. So why has speciation with gene flow become much more common in birds? Well, birds are able to fly, and these birds often exhibit transcontinental and transoceanic movements. And this movement can prevent strict allopatry from actually recurring. And recall down here with our visual diagrams that parapatric and sympatric modes of speciation occur when gene flow is not equal to 0. Why is Beringia an excellent place to study speciation? First, let's define what the area of Beringia is. Beringia is the area that encompasses the region extending from the Russian Far East across the Bering Sea through Alaska and into western parts of Canada and North America. Historically, throughout the Pleistocene, Beringia has experienced fluctuations in climate, specifically glacial and interglacial periods. And as a result, the high latitude flora and fauna that reside in this region have had genetic consequences as a result of these climatic fluctuations, allowing for range contraction and expansion, which results in potential for secondary contact as these populations are being separated and then able to resume contact during these interglacial periods. So jumping back to chapter 1. In chapter 1, we examined 3 subspecies of Holarctic green-winged teal: the Eurasian common teal, Anis creca, the Aleutian green-winged teal, Anis creca, and the North American green-winged teal. We also include the yellow-billed teal, Anis flavirostris, which is a South American sister taxon to fully resolve this complex. And here in the lower right-hand corner, this is a photo comparison of a Eurasian common teal with a North American green-winged teal. And you can see that the 2 subspecies do have phenotypic differences. And recall that this group has previously demonstrated unresolved phylogenetic relationships in both mitochondrial and nuclear DNA. So we can use population genomics to answer our questions that we're interested in. First, we ask 3 questions. What are the phylogenetic relationships of the group? How much gene flow is occurring? And what are the modes of divergence and speciation in this group? To do this, we will use ultra-conserved elements as a molecular marker, also known as UCEs. UCEs are highly conserved regions of organismal genomes that are shared among evolutionary distant taxa. In addition to using nuclear loci, a positive byproduct of producing UCE data is that a complete mitogenome is generated for each individual. So these molecular markers are actually quite useful for the questions that we're interested in. Continuing on to Chapter 1, methods. DNA extraction and sequencing. First, we cut tissues from vouchered specimens at the University of Alaska Museum, or we request loans through the University Washington Burke Museum or Louisiana State Museum of Natural Science. Once these tissues have been cut, we extract whole genomic DNA, which we then send these samples off to our collaborator, Dr. Travis Glenn at the University of Georgia College of Public Health to be sequenced using UCEs as a molecular marker. So the distribution of samples used in my Chapter 1, we looked at the 3 subspecies of Holarctic green-winged teal as well as the South American sister taxon, the yellow-billed teal. In this image, you can see the distribution of the samples that we used. We used 7 Eurasian common teal, shown in light blue, 3 Aleutian green-winged teal, shown in dark blue, 6 North American green-winged teal, shown in gold, and 4 yellow-billed teal, shown in red. So continuing on with the methods with bioinformatics and the UCE pipeline. When we get our raw reads back from Travis, we have to first remove low-quality bases and adapters. And to do this, we used a program developed by Brant Faircloth called Illumina Processor. We used the Mallard to build a UCE reference, which we assembled using Trinity on Galaxy into a reference FASTA file. The reads for our teal individuals were then mapped to this Mallard reference using BWA-MEM. Continuing with more bioinformatics, our bioinformatics pipeline centered on the package Phylucci, again developed by Brant Faircloth. Phylucci identifies conserved orthologous loci that are then used as a reference set of UCE loci. SNP calling was done using Genome Analysis Toolkit, or GATK, which was also used to restrict data to high-quality SNPs. We then used VCF tools to call sites without complete data. And VCF tools was also used to thin the data to be 1 SNP per locus and made biallelic for our demographic model analysis. We then used the R package AVIAN to calculate a principal components analysis, as well as perform assignment tests using discriminant analysis of principal components. For our mitogenomic pipeline, again, mitogenomic sequence data was obtained as a byproduct of our UCE sequencing. And the backbone of our mitogenomic analysis is very similar to that of our UCE data set. Again, with the mitogenomic analysis, we used the complete mitogenome of the Mallard as a reference sequence. The aim of our mitogenomic pipeline was to enable comparison with previous studies that had limited mitochondrial data. For our phylogenetic analysis, we used the program Phylucci again, using their phylogenetic systematics pipeline to generate a single UCE consensus sequence per locus per individual. We then used the program RAXML to reconstruct a maximum likelihood phylogeny of the single UCE consensus sequence. We then used the program SNAP to reconstruct a phylogeny that integrates across all possible gene trees to show the group's history. So essentially an inferred UCE gene tree history. For our mitogenomes, we reconstructed the phylogeny using maximum likelihood methods in the program MEGA. For each of these 3 phylogenetic analysis, we again used the Mallard as an outgroup to give us a confident root. For our demographic analysis, we use the program DADDY, diffusion approximations for demographic inference, to estimate demographic parameters under models of pairwise divergence. We tested 8 models of divergence, which we have a visual diagram of these models shown in the upper right-hand corner. Model A was no divergence. Model B is split with no migration. Model C is split with migration. Model D is split with bidirectional migration. Model E is split with exponential population growth and no migration. Model F is split with exponential population growth and migration. Model G is secondary contact with 1 migration parameter. And Model H is secondary contact with 2 migration parameters, or bidirectional migration. To see if these models were best fit, we used Akaki Information Criterion, or AIC, to see if the model was best fit and that we weren't overfitting the data with a model that had more parameters than what was parsimonious. So once the best fit model was determined, it was bootstrapped to give 95% confidence intervals around these parameter estimates. The best fit model parameter estimates obtained from our demographic analysis in DADDY were used to provide biological estimates of ancestral population size, size of populations, time since split, migration, as well as time of secondary contact if it was appropriate for the model. And again, our parameter of interest is migration. So chapter 1 results. Recall our questions that we are interested in. We are interested in the phylogenetic relationships, how much gene flow is occurring, and what are the modes of divergences and speciation. So the results for our phylogenetic relationships. Our mitogenomic phylogeny, shown in this upper panel, A, we found that the North American green-winged teal, shown in gold, is sister to that of the South American yellow-billed teal, shown in red, to form a clade here. Likewise, the Eurasian common teal, shown in light blue, forms a clade with the Aleutian green-winged teal, shown in dark blue, which is up here, right up here. Our nuclear phylogeny, however, shows something different. Our nuclear phylogeny shows that all the the 3 subspecies of whole Arctic green-winged teal form their own polytomous clade that is sister to Anas flavirostris. So the 3 whole Arctic subspecies of green-winged teal form a clade here, with the South American yellow-billed teal being sister here. And given the low bootstrap support of these nodes in our nuclear phylogeny, it results in a lack of structure. And so to better illustrate these 2 phylogenies, on the right-hand side, I have a sketch illustrating the discordance between the 2 topologies with the relationship of the 3 whole Arctic green-winged teal subspecies within the circle relative to the sister taxon, the South American yellow-billed teal. So the results for our inferred UCE gene tree history, this phylogeny showed the overall same sister relationship shown in our previous nuclear phylogeny using a consensus UCE sequence. And here we can see it is the same overall nuclear topology with uncertain relationships within the 3 subspecies of whole Arctic green-winged teal. The colors in this tree correspond to the first, second, and third most supported topologies shown in blue, red, and green, respectively. And so in this phylogeny, you can see that in the 3 most supported topologies, the South American yellow-billed teal is sister to the 3 subspecies of whole Arctic green-winged teal, while within the 3 subspecies of whole Arctic green-winged teal, there is uncertainty in these topologies. We also tested to see if our mitochondrial topology fell outside of the 95% confidence interval of the nuclear SNAP topology shown here, and it actually did fall outside of this confidence interval, which indicates that the 2 genomic histories are significantly different. So continuing on with the results from our antigen analysis, here on the left-hand side, I have a plot of our principal component analysis and a principal component analysis explains variation in the data. And here we can see again that the 3 subspecies of Holarctic green-winged teal tend to cluster together. However, the yellow-billed teal, the South American sister taxon, does not cluster with the 3 subspecies of Holarctic green-winged teal. Here on the right-hand side, we have our discriminant analysis of principal components results, which is an assignment test of probabilities. And here, this shows a very similar relationship to our PCA analysis, showing the 3 subspecies of Holarctic green-winged teal to form one cluster, while the yellow-billed teal forms its own cluster here. So these 2 plots pair nicely with each other. Continuing on with our results from our data analysis, looking at divergence models and gene flow, these are the visual representations of the best fit demographic models for each pairwise comparison. And the main takeaway from this figure is that each of the best fit models had migration as parameter. And likewise, the level of migration it translated into individuals per generation is not equal to 0 in any of these cases. Even though between the North American green-winged teal and the South American yellow-billed teal, migration is very low, it is not equal to 0. So these are the biological estimates obtained from our best fit data models for each pairwise comparison. And this is a table representation of the previous slide. And the important information is our migration estimates shown in the red box, our parameter of interest. And again, in each of these pairwise comparisons, gene flow, although low in some cases, as in the contrast between the North American green-winged teal and the South American yellow-billed teal, although it is low, it is not equal to 0. So chapter 1 discussion. And recall, we're interested in the phylogenetic relationships, how much gene flow is occurring, and the modes of divergence and speciation. So our first question, what are the phylogenetic relationships in this group? We found discordance between nuclear and mitochondrial phylogenies in the green-winged teal and yellow-billed teal duct complex. Again, looking at our mitochondrial phylogeny, it showed a sister relationship between the North American green-winged teal and the South American yellow-billed teal, with a sister relationship here being between the Eurasian common teal and the Aleutian green-winged teal. This overall topology did agree with previous work that was more limited with mitochondrial data. However, in contrast, our nuclear phylogenies both showed that the 3 subspecies of Anis creca to show an unresolved polytomy, with the South American yellow-billed teal, Anis flavirostris, being sister, again shown here with unresolved low supported bootstrap nodes within the 3 subspecies of Holarctic green-winged teal, with the South American yellow-billed teal being sister. And again, this shows the same relationship over here with unresolved structure between the 3 subspecies of Holarctic green-winged teal, with the South American yellow-billed teal being sister to this complex. So question 2, how much gene flow is occurring? Gene flow was present in each of our pairwise demographic comparisons. Looking at this table again, we are interested in migration, which are our parameters of interest. Migration rates, or gene flow, is not equal to 0 in any of these pairwise comparisons, even though it is low in some contrasts. However, recall that parapatric and sympatric modes of speciation occur when gene flow is not equal to 0. Question 3, what are the modes of divergence and speciation? Divergence and speciation occurred with gene flow in all pairwise demographic analysis. Again, looking at our visual representation of our best fit models and data, the main takeaway from this is that each of these best fit models do have migration as a component in these models, as shown by the arrows. And likewise, the number of individuals per generation is not equal to 0 in any of these contrasts, even though it is low in some. it is not equal to 0. So our chapter 1 conclusions, we found discordance between nuclear and mitochondrial phylogenes in the green-winged teal and yellow-billed teal duck complex. Again, gene flow is not equal to 0 in any pairwise comparison, and therefore strict allopatric speciation is not occurring in this complex. And finally, divergence in speciation occurred with gene flow in all pairwise demographic analysis. And that is the end of my chapter 1. Jumping into chapter 2, again, chapter 2 title is Estimating Movement Rates Between Eurasian and North American Birds That Are Vectors of Avian Influenza. Going into some background for chapter 2, let's discuss 3 main points. What is an avian influenza vector species? Why should we quantify movement of these vector species? And how does this translate to a relative risk of exposure to subsistence users? So background, what is an avian influenza vector species? Avian influenza is a zoonotic disease that is likely to be involved in future pandemics. Waterbirds, such as waterfowl and shorebirds, are asymptomatic carriers of the disease and are a natural reservoir for avian influenza viruses. Many of these birds also migrate to Beringia to breed, resulting in seasonal contact between these lineages. And here we have a photo of Kramer's Field during spring migration, where you can see many different species are using this area as a migratory stopover. So why should we quantify these movements of vector species? In general, wild migratory birds, the number of migrants moving between Eurasia and North America has often been underappreciated when modeling the introduction of avian influenza into North America. Likewise, vast areas of remote land make estimates of intercontinental avian influenza host movements difficult to obtain because you're limited by how many observational field sites you have and what range you have in terms of the ability to get to these remote areas to do these observational studies. However, knowing the degree of these movements between Eurasia and North America is critical to understanding intercontinental transmission of avian influenza viruses. And here on the right-hand side, I have a graphic showing the migration flyways with the Pacific flyway shown in red up here, and Alaska in pink. And you can see that Alaska is the joining place between Eurasia and North America. So it's a great zone of contact between these two migration systems between Eurasia and North America. So how does this translate to relative risk of exposure to subsistence users? Many of these species that are vectors of avian influenza are also common in the subsistence diet of many Alaskans. Avian influenza viral dispersal across Beringia is also not a rare occurrence, and highly pathogenic avian influenza of Eurasian origin has been detected in Alaska populations of waterfowl. Because waterfowl are an important subsistence food for many Alaskans, contact while handling and processing these birds is a potential risk factor to direct bird-to-human transmission. And here on the right-hand side, this is just a breakdown of the subsistence use of migratory birds in Alaska. So back to chapter 2. The overall goal of chapter 2 is to improve estimates of intercontinental avian influenza host movements among waterbird species to better understand the natural host virus movement transmission landscape in Beringia. Given Alaska's proximity to Eurasia, we also contrast these estimates with the importance of each vector species in the Alaskan diets to understand the relative risk. And recall that knowing the degree of these movements in host vector species is critical to understanding the transmission of avian influenza viruses. So in this study, we are interested in looking at host movements, which can further the understanding of the movements of avian influenza viruses. Again, we can use population genomics to answer our questions. And our questions are, how much intercontinental movement, i.e. gene flow, is occurring in vector species of avian influenza? And our second question is, what are the species-specific rates of intercontinental movement with respect to the importance of each species in the subsistence diet of Alaskans? Again, using UCEs as a molecular marker. Chapter 2, methods. Again, we cut tissues from vouchered specimens, which we then extracted whole genomic DNA, which was sent to our collaborator, Dr. Travis Glenn, to be sequenced using UCEs as a molecular marker. The species we sampled in this study are northern pintail, mallard, greater scaup, common eider, common and Wilson snipe. And then we also incorporated gene flow rates obtained by McLaughlin et al. 2020, which include green-winged teal, long-tailed duck, and Eurasian and American widgeons. Overall, our bioinformatics pipeline is the same methods used as in Chapter 1, but the important information out of this is that our thin biallelic VCF file with sex-linked loci removed was used for our demographic analysis to estimate gene flow. That's the important information from our methods. Again, we tested 8 demographic models in DADDY, and the best fit model parameters were used to obtain and provide biological estimates of ancestral population size, size of populations, time since split, migration, and time of secondary contact if appropriate. And again, we are interested in migration, which is our parameter of interest. So next we had to scale movement and obtain harvest rates and calculate a risk factor. So it's important to know that these evolutionary averages of gene flow are very conservative, but as a result, they're directly comparable to each other. So we are able to scale these up. So how do we scale this to census size? First, we obtained population estimates from the U.S. Fish and Wildlife Service and Wetlands International, and then we used the proportion of gene flow in individuals per generation relative to the effective population size to scale our estimates of long-term gene flow up to an approximate estimate of contemporary gene flow by multiplying this proportion to census size. To scale the harvest rates, we obtained harvest rates in Alaska from 3 consecutive years, 2017, 2018, and 2020. To calculate a relative risk of potential exposure to Eurasian origin avian influenza to subsistence users, we estimated the number of individuals harvested of Eurasian origin given the proportion of migration to effective population size and annual harvest. It's important to know that given high variation in avian influenza infection rates, they vary annually, seasonally, and even geographically, we did not add infection rates as a variable. Chapter 2, results. Again, recall our questions. We're interested in the amount of gene flow occurring in these vector species and what are the species-specific rates of intercontinental movement with respect to the importance of these species in the subsistence diet of Alaskans. These are the key findings regarding estimated rates of gene flow. Gene flow was present in all pairwise comparisons. Likewise, estimates for gene flow between Eurasian and North American vector species show high levels of variation of movement between populations. So if we look at this in table form, you can see here in the blue outlined box that gene flow is present in all pairwise comparisons. Likewise, estimates of gene flow between Eurasian and North American vector species show high levels of variation in movement. And overall, dabbling ducks, shown here, showed the greatest magnitude of movements, while diving and sea ducks showed a wide range in the level of movement, shown here. And the shorebird comparison of the common and Wilson snipe showed very low levels of movement with less than 1 individual per generation. So what are our results for the relative risks of avian influenza host species? The key findings are when ranking vector species in decreasing order of relative risk of intercontinental avian influenza movement or exposure, both within the natural system, relative scaled up to census size, shown in this column here, when compared to the context of subsistence users, so the subsistence system, shown in this column, The top 3 species in each context, again, ranking these vector species in decreasing order of relative risk, the top 3 species were greater Scott mallard and northern pintail, although slightly different order. The remaining birds were mixed in their rankings. And finally, common eiders and common Wilson snipe had the lowest relative risks and were equivalently ranked in each of these comparisons. Chapter 2 discussion. Again, recall our questions that we are interested in the magnitude of gene flow, as well as how this relates to a relative risk ranking to subsistence users in Alaska. So how much intercontinental movement, i.e. gene flow, is occurring in vector species of avian influenza? Our study for chapter 2 found that levels of intercontinental gene flow in waterbird avian influenza vector species is highly variable. For example, the level of gene flow is highly variable even within dabbling ducks, and this is a subset of the previous table that we looked at, where we can see that even in dabbling ducks, northern pintails have a very high magnitude of gene flow occurring, with nearly 400 individuals per generation, where green-winged teal have much more moderate levels of gene flow between 23 to 18 individuals per generation. So there's high variability even within dabbling ducks. Question 2, what are the species-specific rates of intercontinental movement with respect to the importance of each species in the subsistence diet of Alaskans? High variation among avian influenza vector species makes it clear that all do not pose similar risks of avian influenza virus movement. And again, this is a subset of the previous table that we looked at, looking at the relative risk ranking of each of these vector species. And so these vector species vary by 3 orders of magnitude in the bird-to-bird natural system, this column here, which is scaled up to census size, and they vary by 2 orders of magnitude in the human-to-bird subsistence system, shown here in this column. And again, this is a subset from a previous table showing here the span of individuals per generation in each. So looking at the big picture, how could subsistence hunting change to mitigate human exposure to Eurasian origin avian influenza? Species with high levels of intercontinental movement might be less desirable to harvest than species with low to moderate levels of intercontinental movement. And so here in panel A, this is previous knowledge without the results of chapter 2, where each of these vector species, there's no relative risk ranking, and we don't know the magnitude of gene flow occurring between Old World and New World lineages. However, in panel B, with the results of this study, we can see that some vector species, such as the greater Scott, have a relatively high risk ranking, while American widgeons had a very moderate level of risk ranking relative to the amount of movement between Old World and New World lineages. And likewise, common eider had a relatively low risk ranking. And as a result, this information here could allow subsistence users to selectively harvest vector species that pose a lower risk of potential transmission of avian influenza. Chapter 2 conclusions. Overall, our study found that levels of intercontinental gene flow in waterbird avian influenza vector species is highly variable. And this high variation among these avian influenza vector species makes it clear that all do not pose similar risks of intercontinental virus movement. And that is the end of my chapter 2. Finally, I'd like to acknowledge everyone. A big thank you to Dr. Kevin Winker, Dr. Naoki Takabayashi, and Dr. Devin Drown, who provided helpful comments and feedback on earlier drafts of the manuscript. I would also like to thank Simca Gillette, Katie Collier, Jessica McLaughlin, Rebecca Cheek, Jack Withrow, and Dr. Kevin Winker for their invaluable help and support throughout my time at the University of Alaska Fairbanks. And with that, I would like to thank everyone for coming. And now is the time for questions. All right.
Hi, this is Ed Murphy. I don't know if you can see me. Can you hear
I
me?
can see you. Yep, I can see and hear you.
Okay. I was just wondering if the difference in your mitochondrial and nuclear phylogenies is due to a difference in phylopatry of males versus females, that females might be moving less?
Yeah, so that's actually a potential hypothesis that we were considering, is that male-mediated gene flow could be the result of this mitonuclear discord, because males and females pair up before the breeding season in ducks, and the males follow the females.
Right.
So that could definitely be a reason why there is mitochondrial and nuclear discord in this group. Thank you.
Thanks.
I have a question to piggyback off of that. I was curious, given that discordance, what would you recommend to help try and solve that phylogeny?
You mean like
future steps?
Oh, future steps? I think that, I mean, our UCE data set is pretty comprehensive, and we have a lot of loci that we're looking at, but if you could do whole genomic studies, that would certainly, I think, even further resolve, or at least our nuclear phylogeny, since we did look at whole mitogenomic DNA. Any other questions?
I've got one. So first of all, really great talk. My main question, looking at the results, particularly with Nimia, and considering both that polytomy that you're getting and where it is geographically, kind of out on these islands between the ranges of Kreeka and carolinensis, have you considered the possibility that taxa, like the reason you're getting the polytomy and we're seeing the patterns that you're seeing, is that it is actually of hybrid origin between Kreeka and carolinensis?
That's actually an interesting thing to bring up. So one thing that I don't think I mentioned is that the Aleutian North American green-winged teal is a sedentary population out there. And so it's possible that's something that you could consider. And in our mitochondrial results, I don't know if you saw, but there was actually one individual of one Eurasian common teal individual that actually was within the North American green-winged teal clade, which could be of hybrid origin. So that is an interesting question.
Yeah, I got another question, not related directly to that, but that same chapter. I don't know how many loci you've got on the Z chromosome, but did you consider running the Z chromosome data separately to get at that question of whether sex-mediated, differences in dispersal by sex might be driving that? Because you'd expect if that was the case, you'd see that also reflected in the Z, but I don't know if you have enough loci there.
I didn't actually consider that, but that's an interesting analysis to try to look at the loci on the Z-linked chromosome. That is interesting. Yes.
Cool. Yeah, I thought of that because that's something that I got suggested on my dissertation comments recently.
Okay. Yeah, that's an interesting thing to think about. Yes. Thank you.
So, but yeah, awesome job.
Thank you.
Hi, Fern.
Hi, Derek.
Great talk. Yeah, and I was just thinking more about that mitochondrial nuclear discord and wondering, do you think the lack of monophyly for the green-winged teal in the mitochondrial data might be due to like retained ancestral polymorphism, or do you think an alternative might have been some kind of mitochondrial capture or hybridization more recently?
It could be ancestral hybridization that is causing that in the mitochondrial DNA. It could be, yes. Does that answer your question? Or
Yeah, thanks. Yeah, that's another possibility, I guess, is ancestral hybridization. Yeah, it's kind of hard to tease all that out. But
yes.
good. So, and also just curious, maybe I missed it. How many sort of base pairs was that UCE data set? I imagine it was huge.
Yes, it is huge. And for our chapter 1, I believe we were looking at like 1900 loci
Oh,
is
wow.
what we were using.
Okay. Thanks.
Uh-huh. Any other questions? And again, if you have questions after this talk, feel free to contact me through email as well. Or if you would like a PDF of my slides, I'm happy to send that to people.
I have a somewhat more of a cultural ecology question, perhaps, with chapter 2 and how a lot of the subsistence harvesting was dabbling ducks and how you could reduce your risk by going after eiders instead. I was wondering if there's like a specific reason why a lot of the subsistence users have been kind of seemingly biased towards a certain species. Is there like a cultural reason, or is it like an ease of, it's just easier to shoot scops or something like that?
Oh, so in that diagram, I may not have made it clear, but so I took like the top ranked species in terms of risk ranking. So like the top risk ranking was scops in our results. And then a moderate risk ranking was widgeons, and then a low risk ranking was eiders. And so I used that as an example of potentially having subsistence users, giving them the option to selectively harvest vector species that posed a lower risk. So that diagram itself is a reflection of our risk ranking results. Does that make sense? Does that answer the question?
Are you coordinating with any tribes to try and like disseminate this type of information so that they are so that they have these types of guidelines with them?
Not yet, but our harvest rates were obtained from the Alaska Department of Fish and Game Division of Subsistence. So as we move along with chapter 2, that would be an aspect that I'm interested in pursuing, is providing subsistence users with this information to allow them to have the knowledge to selectively harvest these vector species, at least giving them the knowledge so that they can make that decision.
Mm-hmm.
Are there any more questions for Fern? Speak up, we've got some time.
I guess with regards to selective harvest of species, are there, am I coming through?
You're a little crackly, but you can go ahead and try. I'll see if I can.
Okay, sorry about that.
That's okay.
But with regards to subsistence hunting, are there any guidelines with which people could, even if you're harvesting a high risk bird, are there any protective mechanisms that you could put out there to lessen the risk of contracting avian influenza in processing and handling the birds, even if you do have to take a higher risk species?
Mm-hmm. Yeah, so actually the Alaska Department of Fish and Game and even the U.S. Fish and Wildlife Service does put out guidelines for processing and handling of wild game. And so some of the recommendations that they have are to wash your hands and to not, lick your fingers while you're actually processing these birds. Just general common sense precautions is like their main procedures for mitigating that potential risk of transmission. So they do actually put out guidelines for how to mitigate that risk. Mm-hmm. Any more questions?
Well, if not, let's thank Fern one more time for her presentation, and a few of us will get together in a smaller Zoom room for further questions.
Okay.
Thanks, Fern.
Thank you. Thank you all for coming.
Great job, Fern.
Thanks. Bye, guys.
Bye. Nice job.
Bye, good to see you.
Bye, Rebecca.
Bye.
Bye, Ram. Okay. Let's see. So you have the meeting, the private defense set up, right, Kevin?
Yes, and I think I've got that set up for 11:15.
Okay.
Is that
So, yeah.
okay, Naoki?
Yes, yes, yes. Okay, let's see. There's another link. Oh, yeah, yeah. Okay, yeah. Sounds good.
Okay.